Hi, in this video, I'm going to talk about glycogenesis. Let's break the term. Glyco means glycogen here and genesis means formation. So basically this means glycogen formation. So when we take a meal food of carbohydrate, it's get broken down into glucose, and that glucose have to be stored somewhere inside the body as a energy reserve. The glucose is usually stored in the liver or in the muscles, and the process by which glucose get converted into the glycogen, and that means a storable form, is called glycogenesis. So let's see step by step how this glycogenesis process occurs. So here we have glucose. Glucose is our substrate for glycogenesis. So glucose would be converted firstly to glucose 6 phosphate. Glucose would be converted to glucose 6 phosphate, and this is a phosphorylation reaction, and this reaction would be catalyzed by hexokinase enzyme, which is a very important enzyme in this pathway. Next, what would happen? Glucose 6 phosphate, now it would be converted into glucose 1 phosphate, and it's an isomerization reaction, and this reaction is catalyzed by phosphoglucomutase. This reaction is catalyzed by phosphoglucomutase. So now if we talk about the structure of the glucose, glucose somewhat looks like this. So this is the basic structure of glucose. So here is the 1 2 3 4 5 and sixth carbon. So in the sixth carbon, what would happen in the next step? Glucose would be phosphorylated in its sixth carbon, and it would form glucose 6 phosphate. So let's see. So here in the sixth carbon of the glucose, here we would attach a phosphate group by this hexokinase enzyme, and this is called glucose 6 phosphate. And now what happens? Glucose 6 phosphate would be converted into glucose 1 phosphate, and this glucose 1 phosphate is actually a isomer of these glucose 6 phosphate, I say. After getting converted into glucose 1 phosphate, it would be converted into UDP glucose, and the reaction is catalyzed by UDP glucose phosphorylase. This is catalyzed by UDP glucose phosphorylase. So now it would be converted into UDP glucose, and UDP glucose would look like somewhat. So this is the structure of UDP glucose when a uridine moiety is attached. And UDP glucose is very important for addition into a preformed glycogen polymer. So we have a preformed glycogen polymer. We would denote it as N, and now it would attach with UDP glucose, and ultimately it would form glycogen, and it would increase the chain by one residue long. So this would be the final reaction. So now if we talk about how this preformed glycogen polymer appears, so we should talking about a protein named as glycogenin, and this glycogenin protein is actually a dimer. So this glycogen protein has two subunits, subunit A and subunit B, and at the terminal position it has a tyrosine group. With this tyrosine group, some glucose molecules, some glucose moieties are actually attached, and these glucose moieties are with these glucose moieties, further attachment of other glucose moieties takes place. I mean, UDP glucose directly attached to these moieties and elongate the chain. That is how it occurs. So the chain gets elongated continuously, and this is the basic reaction scheme. So next we would talk a little about the regulation. We would talk about regulation. So one enzyme I want you to remember is in this step. This step is catalyzed from glycogen plus UDP glucose, and it forming a glycogen N+1 residue. This step is actually catalyzed by one very important enzyme, which is a rate limiting enzyme for this glycogenesis step. This is glycogen synthase. And this enzyme is very important because it is the rate limiting enzyme. So this is the rate limiting enzyme in this pathway. So this rate limiting enzyme could get allosteric modification, so that we would see how this rate limiting enzyme get allosteric modification. So glycogen synthase generally have two forms. One is it has glycogen synthase in A form, and glycogen synthase in A form is not phosphorylated and it is active form. Another form is glycogen synthase in B form, and glycogen synthase in B form is inactive because it is the phosphorylated version of the enzyme, and in a phosphorylated state this is inactive. So it is phosphorylated, as you can see, and this is not phosphorylated, and this is active state. So the switching between these active conformation and inactive conformation would ultimately regulate this pathway. So let's see how this pathway take place. So now what happens? So from inactive state to active state, switching is catalyzed by a phosphatase enzyme, phosphoprotein phosphatase, and this enzyme is regulated by other hormones such as insulin. So what happens? Insulin actually triggers the activity of phosphoprotein phosphatase, and thus inactive form of glycogen synthase get active, and ultimately the glycogenesis reaction take place. Glucose get converted into glycogen. But glucagon and epinephrine on the other side allosterically hinders this phosphoprotein phosphatase. Glucose 6-phosphate also negatively regulate phosphoprotein phosphatase, and on the other side the conversion of active form into inactive form is triggered by GSK, glycogen synthase kinase. So when glycogen synthase kinase is active, what will happen? Glycogen synthase would become phosphorylated, and ultimately glycogen synthase would be inactive. So insulin actually inhibits this glycogen synthase kinase. So insulin inhibits glycogen synthase kinase, so the active form doesn't get converted into inactive form. So in presence of insulin, the glycogen synthase A form, that means the active form, takes part in the reaction, and it ultimately converts glucose into glycogen. so that blood glucose level could be lowered and the glucose could be stored inside our liver for future purposes. Hope you enjoyed the video. Please like and subscribe.